All right, everyone. Let's go ahead and get started. This is the session on utilizing network imaging to personalize and optimize neuromodulation in translational cognitive neuroscience. I am your session chair, Roy Hamilton. I am a behavioral neurologist at the University of Pennsylvania, the director of the Laboratory for Cognition and Neurostimulation, and the director of the Penn Brain Science Translation Innovation and Modulation, or Brainstem Center, again at the University of Pennsylvania. So the rationale behind this session is really that it aims to address the notion that a lot of what we think of as advances in neuromodulation can be attributed to advances in our ability to conceptualize and represent the brain with respect to complex dynamic networks, to our ability to get a finer sense of brain circuits and how those brain circuits are integrated into larger brain systems, and then use that understanding to guide transcranial magnetic stimulation and other neuromodulation modalities in order to be both more personalized to the individual and also optimized with respect to the effects that we hope to elicit from each individual, right? And so the two talks we have today, both by faculty at Drexel and Penn, but also affiliated with the Penn Brainstem Center, really highlight this interplay between our ability to understand brain networks and use that understanding in order to guide interventions that involve neuromodulation. So let me just say a word about how the session is going to run. I hear some background noise, so I think it would be helpful if everyone else who's not a speaker quickly muted themselves. I think that would be helpful throughout the session. So let me say a couple other rules about the session. Each talk is going to be about 15 minutes long. After each of these two talks, we'll have about a 5-minute period for question and answer. You can type your questions into the chat box, if that might make things easier, and I can read them out. I might not be able to address all questions to the speakers in that 5-minute window. However, at the end of the session, we'll try to get as many questions as we can answered, including ones that were addressed or ones that were addressed to speakers after their individual talks that we didn't have time to get to. All right. And so with that, it is my pleasure to introduce our first speaker, John Medaglia. So John Medaglia is an assistant professor of psychology at Drexel University, where he runs the Cognitive Neuroengineering and Well-Being, or CogNEW Lab. He'll be talking to us on the topic of integrating brain network anatomy and function to enhance cognition with neuromodulation. Take it away, Dr. Medaglia.
Thanks, Roy, and for chairing the session. I hope everybody is finding themselves as well as possible in the circumstances. I sort of took this invitation as an opportunity to consolidate a lot of what we've been doing in the lab and with colleagues, especially at Penn and also Georgetown University. And I thought it might be interesting to sort of cast a 30,000-foot view on some major themes in neuromodulation, and then sort of land on a couple of salient examples that had either been recently published or are being prepared to be published now. And hopefully that sets a context for some of the specific applications that Desmond will talk about in a bit here. So I don't know if I was asked explicitly, but often with this conference, we're asked disclosing relationships. Our primary funding all comes from federal and university sources and a charitable donation from Starfish Neuroscience. Today, I really want to just be focused on a couple of claims here. I think I want to orient us to the idea that network anatomy varies across subjects and can guide network function and cognition. So I'm going to take a sort of ground up view on the way that anatomy might be wired in general for individuals and then differ across individuals, and how we might find that interesting, predicting both what we see in functional neuroimaging and then indeed cognitive outcomes. I think that in general, as you'll see as a theme in Desmond's work as well, that predicting neuromodulation outcomes could improve, sort of generally speaking, with person-centered network neuroscience. And I think that by viewing these problems in this way, I think we can identify some fundamentally new modulation practices. So you'll see what I mean by fundamentally new, hopefully through a couple of the examples today. So I think a lot now about this problem of neuromodulation when your N equals 1. So like many treatment designs, when a person presents to a clinic, we have education that supports people making general claims about what it means to have a disease and how we diagnose that. But in the end, every clinician is confronted with the problem that, well, I have to treat this person in some way. And as we all know, the treatment responses vary. And then this is a challenge both in terms of how we diagnose and then prognosticate what happens with that person. So fundamentally for neuromodulation, those outcomes are some interaction between characteristics of the person and characteristics of the neuromodulation. And I think that for not just myself, but many, including Roy and Desmond in the room, have been pointing out that precision neuromodulation is appealing, but it's not obvious that precision pharmaceuticals is the best model for this. I kind of want to orient us to the idea that we might find this concept of precision neuromodulation to be appealing, but it differs in a few interesting ways from pharmaceuticals. And one of the ways that I tend to like using a network representation to think about this is that if you're designing some kind of pharmaceutical to interact with neurotransmitter process or otherwise, we fundamentally have something at a very low scale of organization. And not encoded often in that neurotransmitter is the idea that the scale has dynamics that might not be observed at that lower level. So to make that an abstract claim a little bit more concrete, you have many different regions in a brain network, each of which is comprised of many different components, and those somehow have to interact with one another to do things like promote cognition and behavior. But it's not always obvious that any particular component of that structure is what's driving that particular behavior. So we have a special case in two forms. One, what is the role of the elements in the context of this broader network? And each of those could be sort of configured in their own way. And then two, that network is reconfiguring to support different kinds of states as we navigate different behaviors and cognitive function. So the broad challenge in this sense is, how do you take something like this general network intuition and ask smart questions of the behavior to predict outcomes, and by virtue of that, enhance the odds that you can improve function? So rhetorically, I ask myself every day we're doing this work, can we in some sense move from off-label and to on-target? So we're using neuromodulation these days for many different things, but here I find it helpful to think about a simple model often supported by the RDoC initiative through the NIH, where if we view neuromodulation and we have some kind of outcome of interest, and the outcome could be hours or years, we often are thinking about, or not, some pathway to the treatment to the outcome. And the idea in the long run is that if we can find mechanisms that mediate outcomes, well, now we can design classical randomized control trials and see if the mechanism-informed treatment outperforms the no mechanism or other mechanism-informed treatment. And hopefully this scaffolds us to better opportunities and outcomes in the long run. So I think this model is, there's no fundamental reason to question that logic at its purest form, but then the details are, well, what mechanisms ought we care about and how and why? So I just want to orient us to maybe one view on this that we wrote about recently, that there seem to be two cultures that are consistent with statistical cultures in general when approaching this problem. And you might call those interested in statistical fits versus those interested in mechanistic models. And this first style is, we have maybe a task that we're doing or some clinical outcome. You have some observed performance, you'd like people to be faster or have less of a clinical score. And that distance measure is really what you're trying to enhance with neuromodulation. Can I make the person better? And you might go ahead and say, well, we have a lot of data we can stack these days with genotypes. We can know a lot about your background socially, demographically. You can get all kinds of imaging data of structure and function, and we might want to represent those as different sorts of data elements. Here I'm using a network intuition to show that each of these could be its own measure, but you could also ask about, well, how are all of these linked together in some sort of network, and then define measures over those networks of elements to do things like predict outcomes. And that can often look like something like people taking neural network models, random forests, support vector machines, and so on. And ultimately, we're trying to predict, well, what's this delta from where I was before neuromodulation and where I was afterward. And we might be able to make some conclusion about these associations and how they come together to drive those outcomes. So that's what we'll call statistical fitting. Style 2 is what we might call mechanistic models, where we have the same objective. We're caring about this distance between where we were and where we're going. We still might be concerned with the same data elements, but we have some sort of order by which we care about them. So we might imagine that before the person comes to the clinic, they have some genotype, some social status, they have some particular configuration of their brain network anatomy in its entirety or in its details. There might be some way to abstract that as a network representation, as a feature of the system itself, not just associations among features. And then we can do things like say, well, we know something based on cognitive neuroscience about maybe how a cognitive model should work in this brain network, and then how maybe that relates to the neuroscience of that brain. And so this is what we'll call cognitive neuroscience in the context of network neuroscience. And we can use the many traditional techniques of which you will be familiar with about how does the brain oscillate, what are sort of the event-evoked features of doing different things in different conditions. And then ultimately saying, well, if we knew where we were before, can I go ahead and stimulate somewhere in that model to make a prediction based on these models about what would happen in these responses, then change it to some outcome that we can predict. So the general observation I want to make here is that in many cases, even if the statistical fitting case is available to us, we often have a lot of knowledge and strong priors about what might or might not seem more plausible in the first place. And then indeed, we can be competing basically between our sense of mechanism and our statistical fits, and these should drive one another to improvement in the long run. So I just want to briefly then remark on maybe one general way to think about this sort of in a ground up way for the N of 1. And one way we've written about this is that basically you can think fundamentally that there's some white matter backbone to the brain. So different regions need to communicate with one another over many pathways. And these are quite complicated, but we can represent them mathematically in a relatively straightforward way in modern days. We can use, and I'll talk a little bit about network control theory as one version of thinking about this, where you can then make some simulations or predictions about, well, how does this particular node drive function over that network or a subnetwork? And we can use tools from engineering or otherwise to make estimates about that property. Properties might predict then how dynamics in the brain move, where by dynamics I basically mean what you would predict to see in something like the fMRI machine or EEG evokes data. Cognitive control functions is something I'll talk about today. And then ultimately, if we can grasp the concepts, linking this anatomy through the dynamics to the cognition of the brain states, well, there should be some class of these states that helps us enhance our translational benefits over time. Cyclical, where the better our data get on the way in and the better our models get on top of those data, we have better tuned predictions that can then sort of winnow out, well, what's most likely to be maximally effective in the long run. So this is an iterative process and concept. So what networks really add, at least in one account of this, is that we haven't thrown out anything we thought we knew about the brain. We still think there are different cells in different places, maybe doing different things. But what we really get to do is say, well, there are different scales to that organization. And it's sort of trivial in a network sense to say, well, I could represent the single neurons as some complex network in addition to those regions or vice versa, and then think about how these networks are configured and reconfigure to do things like organize the function you might see. So many of you might be familiar that you can go and take a lot of fMRI data and parcellate it into major networks and associate those with behavior independently. What is interesting to note here is that that functional organization is basically a composite of many different regions communicating in some particular way. Now that function, as is maybe not as often explored, but increasingly so, is in some ways very much related to this anatomy, in other ways not. And one of the challenges I think we're confronted with is, well, putting that all together, if I'm in the end caring about some functional difference across people and maybe in response to neuromodulation, how can I tie these things in a way that we can cohere and explain to ourselves and then use for treatments? And so the brief example I'll just mention here of one way of thinking about this is we can do things like, say, have people focus on simple shapes in the scanner, ask them to switch back and forth between tell me the big shape, a little to see here are there little shapes like circles, X, and triangles and squares. And then the color basically cues people. Well, tell me either this large triangle you see or these little squares that make it up, or tell me the little X's that make up the circle. So what this does is it causes a person to have some conflict. You have to suppress looking at the level that you shouldn't look at. And people are surprisingly different at even these basic tasks. So as you'll see in a moment, some people are twice as fast as others, even in healthy young samples, at how they switch between these levels. So we asked the question a couple years ago, well, if I care about how this looks in each and every one of us, and I care about how that organizes the function, I have a fairly complicated problem on the face of it, where what do I focus on? So do I focus on every region, the region connecting this and that? What's that pathway? Well, there are now little triplets of pathways, so how do I deal with those triangles of regions and so on, until you can just appreciate the entire brain. So when we see a pattern like something in an fMRI machine, when somebody's doing the task, we ask the question, well, how does that person's anatomy align that person's function? And how is that in some sense an alignment across the entire complexity of this? So using some tools from engineering known as graph frequency transformation, we can actually decompose this signal into what's called the spectrum of this brain anatomy. And then as a function of that spectrum, asked, well, overall, how aligned is that signal? So it looks something like this. I'll spare some of the math here and just give you a visual example where the math basically decodes, if we have a little toy network and we have this sort of bow tie, and if your brain only had 6 regions and these few edges connecting them, where these blue edges are the anatomy, you could say, well, it would be very much aligned if the signals tend to be up when they're well clustered together and down when they're clustered together. And it would mean that it's unaligned, or liberal, as we termed it, if it's sort of devoid of that information. So I'm kind of, saying, well, I'm going to not stick really to that clustering organization. So we can define a measure basically using all of the data in the brain, both the anatomy and the function, through a single number that captures how aligned or how liberal the person is. And we were sort of generally interested in, well, if you need to switch really efficiently, it might make some sense in which the brain signals aren't sticking so much. So we actually expected this to be more predictive of people's switching ability. So we went ahead and did exactly that. We decomposed the brain signals, and then we said, well, let's find the relationship between these aligned signals, and we can plot them out in terms of where they tend to inhabit the brain, and find that it doesn't really predict this measure of switch cost, how rapidly people can switch between the levels. However, when we looked at this liberal signal, so again, this is how unsticky or how liberated from the anatomy it is, we found that this was much more strongly correlated with performance. And it was sort of interesting to us that it actually turns out that if these signals are really liberal, they actually benefit from a little bit of alignment from the anatomy. And for those people that have these sort of free-floating signals that are sort of tracking a little bit with the anatomy, sort of flowing over that from moment to moment, they're maybe 2 to 1 as fast as the people to which these signals are really sort of randomly ordered over the network. So in other words, a little bit order goes a long way, and we can represent that in a single measure, integrating the anatomy and function of the brain, accounting for all the complexity imaged here in this picture. So to a cognitive neuroscientist like me, this is exciting because you sort of find a single number representing a lot that correlates with an outcome, but it doesn't quite do enough for me in terms of how we make people better with neuromodulation. So recently, especially with Roy Hamilton's group and us, we've been asking questions like, well, if you notice on this previous slide, I'm kind of just letting the whole brain talk to me at once, but we can still care about, well, how does that region and how does that edge, et cetera, participate in the way that I behave and the way I respond to neuromodulation. We still have this problem of sort of the figure in the ground. Where should I look in this network? So you can sort of find a compromise where you say, well, how can I look at a single region and the context of the whole brain network properties? So I'll just give you one example for how I'm thinking about that in controlled language processes. Now, a controlled language process can mean many things. We tend to look at tasks like this, where we'll have a subject sit down, we'll get them comfortable outside the scanner, and we'll just give them a sentence and we'll say, Vic asked her to repeat what she had. And most people will tend to say said or something very similar in response to that. And the reason is that when you have a sentence like that, your brain is already kind of calculating as you read it, oh, this is a really appropriate word that I might expect to say at the sentence. Here, you can see something like, few had the nerve to take the needed, and then what? People might say step or action or time, each of which might be a little more weakly associated with this, and each of them is about equally appropriate, so people will actually have a harder time choosing this. They slow down, they occasionally make more errors. The same can be said for simple stimuli like nouns and then verbs, where in this verb generation task, we say, okay, I say move, what do you do with that? So if I see a movie, I watch it. However, what do you do with the crane? Well, you might lift or carry or attach and so on. So basically these tasks are a nice manipulation of how easy it is to select among these different competitors and how easy is it to retrieve those competitors we associate. So what we did here was we took an informative inhibitory TMS known as continuous theta burst stimulation. We target a region that in advance we think is involved in these processes, and we basically try to inhibit some of that activity. And then we parcellate the underlying anatomy here. So for every subject, we take their white matter, we segment that out with diffusion tractography into a parcellation where we can sample the brain down to regions we know and care about. And then we can represent that as a network. And so here you're just seeing each little green dot here of a different intensity is how strongly connected every region pair in this is to one another. And we can do that across all of the regions in the brain. We can then identify how well clustered are different pieces of that in each person. So if we're looking at anatomy, unsurprisingly, we see things that are sort of adjacent in familiar lobes tend to be really highly interconnected. So frontal cortex tends to really connect to frontal, temporal to temporal, and so on. But then that varies a little across subjects. So we can find that in each person. And then we can do things like say, well, let's go back to that region, like the IFG, and say, what's your role in the context of the network? Something like a property known as boundary controllability says, all right, well, if your IFG has a big role in mediating the different colors over here, I'm helping red talk to blue to yellow to green, it's going to have high boundary controllability. It helps communicate among these modules. A concept like modal controllability, you'll notice, is much more on the periphery. They're sparser. They're not as strongly connected. So this is often something within a system that's sort of talking to nearby neighbors weakly. It deals with specific and harder to reach states and concept. And what we can do is then project that out across all subjects in the sample, and we can observe and have shown that that differs across individuals' brains at the anatomical level. So some people have quite strong boundary controllers at IFG or weak modal controllers and any combination thereof. So what we did here was we said, all right, well, are all control demands created equal? And what was sort of interesting to us in these tasks is that the context of the task clearly matters for how hard it is for you. So if you have to finish a sentence, it's actually harder for you to select among appropriate options. And we can kind of know this intuitively when we have a conversation where if you have many possible options of what to say to somebody, you don't know, in fact, what you want to say to somebody. And so sometimes we have increased selection demands when dealing conversational sentence completion. A sort of strange task, like how often are you asked off the street, here's a noun, give me a verb. Well, people actually find it a little harder just to retrieve the words in the first place as it becomes harder to retrieve them. So here we sort of anticipated we might find more sensitive sentence completion effects because of the selection and retrieval connects effects for verb generation. And in a nutshell, because I'm bumping over my time here, so we basically established a link in the study between the role of this language region and context-rich semantic demands. And so what I mean by that is when we look at this region and then we pull out these properties, boundary but not modal controllability is linked to how hard people find it at baseline to find the words that they need to say. And so what that might look like is in this blue line, when that boundary controller is much stronger, you have about the same effective selection, so getting to that right word you want to say. Whereas over here in sentence completion, boundary controllability sort of deviates. And it actually turns out that if this region is much more important for integrating these modules, tend to actually be slower the harder the task is. And one way we are thinking about that now is that basically what happens is as this region has more and more demands at intermediating processing across this network, it becomes harder and harder for it to parse among the hardest stimuli in the data. So we might expect, in other words, that these people really struggle with, they're trying to get across a hard concept. They can't quite kind of find the words. They're going to especially struggle with tasks like that. We care about this clinically because we know for folks like those that have suffered aphasia that they're going to have language problems. And so they're going to have difficulty saying and finding the words that they want to say. So this effect is sort of interesting in that perspective. There's a lot going on in the behavior on this slide, but I want to point out basically that we then took this property and said, all right, well, if they're having some trouble in sentence completion as a function of that property, is that also explaining part of how they respond to TMS? And the short answer is we have a little more evidence in favor of that for the sentences than in verb generation. And basically what we see is that as people tend to do this in a sham condition for sentences, they usually slow down. And this is maybe a semantic satiation effect. As you continue to have somebody parse a lot of sentences and think about what to say, you're activating this language network. There's competition among the things that you might say, and people actually slow down more than you expect from what you might get from practice. What happens with the TMS is that basically we alleviate this effect. So here, if the boundary controller is stronger, we're alleviating some of that semantic competition. The person gets faster again. However, if the boundary controller is weaker, so again, not as strong in this property, then that person is still going to be susceptible to this semantic satiation. They're going to have trouble getting the words out the more they're doing the task. And so what we've been doing now basically is we have a pilot whose data we're processing in individuals who actually have had strokes to understand if this property can help account for how the person might respond to TMS. So to wrap up here, open questions that we have are, does the brain anatomy guide the dynamics in the brain? So notice there we were just asking about anatomy with a really simple model, but we might predict the function based on some of those model predictions, and we'd like to see that, and that it predicts language and cognitive control. Are these simple, reliable measures of this target's intermodule connectivity linked to high-value clinical outcomes? So again, I'm excited already that we're getting a little bit of this link at this node, but we're curious to see if we can make patients with aphasia better. And finally, and prospectively, we wrapped up a pilot recently that we're working on the data now, where we can go ahead and then try to assign subjects based on those network roles, like boundary controllability, and then see if the outcomes change compared to conventional targets. So with that, I think I'm a bit over time. So thanks to Roy and the organizers, and I'm happy to field any questions.
Great. Thank you, John. In the interest of time, I think I'm just going to read the two quick methodological questions that appear in the chat box. John, you can see them as well, but I'll read them out to everyone. So first question, John, was the TMS applied on the left or right hemisphere? And what type of population, healthy or clinical?
Great question. Sorry, I glossed over that. So in the particular data that you just saw, we were stimulating in the left IFG, which is traditionally associated with parasylvian language networks. And we were looking predominantly in healthy subjects here. The data I mentioned at the end are subjects with aphasia who received basically the analogous strategy to the right IFG, which often is thought to accommodate function after the injury, or a region that we picked based on the network role somewhere in the bilateral language network. So we're excited to see the outcome of those data now.
Great. A second methodological question. How were the stimuli presented, visually or auditorily? Depending on that, there are implications from a reading or spoken language perspective. Also, did you look at correct versus incorrect responses in the context of suppression of incorrect responses?
Great question. So, so far, all of these tasks are presented visually. So they get the sentences in little segments and then complete it, or they get the single word on the screen. And I agree that that is a huge issue for sort of naturalistic and ecological context. So I'm hopeful that we'll find some support for that work after this study. Sorry, Roy, can you remind me the second part?
It says, also, did you look at correct versus incorrect responses?
Yes, so what's a little challenging in this particular data set is, and I think part of this is because we give them the visual stimuli, they're very accurate. So people are usually pretty close to ceiling. They're hitting at least 95% accurate in most cases. So we've mostly been looking at response times so far. We're going back now to see if there are actually some cases where there are very pronounced issues with accuracy, even in the healthy sample. In the clinical samples, we're looking both at response time and accuracy because the aphasics are far less accurate.
Fantastic. All right, we'll go ahead and move on to our second speaker. So Desmond, if you want to go ahead and screen capture or
stop mine.
Your screen. So it is my pleasure to introduce Desmond Otis. Dr. Otis is an assistant professor in the Department of Psychiatry at the University of Pennsylvania. He is the director of the Otis Lab, but he's also the associate director for the Penn Center for Neuromodulation in Depression and Stress. And he is going to be speaking to us on interleaved TMS fMRI for causal mapping of cortical subcortical connections towards novel personalized treatment targets. Dr. Otis.
Great. Can you see and hear? Okay.
Yes and yes.
Okay, thanks. Thank you for the invitation, Roy, and it's nice to see some friends in the audience, so thanks for attending. I'm going to talk about interleaved TMS fMRI. I spend a lot of my time working on that and methods for improving that. One of the motivations is making clinical treatments work better. So many of you are aware that a lot of the depression treatment with TMS still happens with the 5 centimeter rule, which is find a motor response, go 5 centimeters ahead and stimulate there and hope you're in the DLPFC. So that there's a sizable minority of people that you definitely shouldn't call that the DLPFC. You're in other territories, and so we want to do a better job at figuring out where we want to stimulate and individualize that better. If you look into the fMRI literature, there's a similar problem, which is that if you just look at group maps, like you see on the left side for a common working memory task, you see a certain pattern of activation. But then if you look on the right of how many actual people have strong activation in those particular areas for that same contrast, it looks a little bit different. In most cases, a bit smaller, and the shape changes a little bit more. And so the point here is that if you just take a group map, even if it's derived from a nice meta-analysis or some a function that you care about a lot, apply that at the individual level without collecting any individual functional data, you may be not getting the best sort of target for that individual. And so making fMRI more individual in terms of fingerprinting and all the various ways that people in the field are trying to improve applications to individual subjects is very relevant to doing TMS and individualizing it and hopefully making it work better. This is from Mike Fox's work. Hi, Mike. So the group map, this is kind of re-emphasizing a similar point, but specifically looking at a common depression treatment target of the subgenual cingulate. If you look at the group map here with the black circle looking for a peak where you might want to stimulate. If you apply that to any individual subject, you might be doing a pretty good job. Well, like with the center brain with that black circle, but when you try to apply that to another individual subject, you might actually be hitting the circuit, pointing it in a direction that's the opposite of what you intended. So that's just re-emphasizing the importance of having actual data from each individual and using that as a guide for how you might apply stimulation. So the way we do this is you can take any map. So you can take a structural map, a diffusion map, a functional map from a task, resting state, and we can do neuronavigated TMS, which is taking an MRI of that participant, using a neuronavigation system to map their scalp to their brain image, and you can overlay, like I said, any other kind of image you want to on that person's brain, and you can move the TMS coil around on the scalp and know what underlying brain area you're stimulating. So that offers a lot of fun possibilities for trying to map functions and modulate them with individual participants. What we do in my lab is interleaving TMS with functional MRI recordings. And the way this is made possible is because an fMRI response to any event is actually fairly slow. So you're not getting the peak from that fMRI response for several seconds after any event that you might have in the scanner. So that allows us to apply single pulses of TMS and then start the subsequent recording of the signal, and we're able to capture what that TMS pulse does throughout the brain. And so what that looks like is we have this brain target, we stimulate it, we interleave the single pulses with fMRI readouts, and we can make maps, causal maps of information flow from that place where you stimulated throughout the rest of the brain. So that really gives you some sense over which areas kind of push the other ones and have potential to move distributed brain targets throughout the brain, so you can develop a map like this. We started doing this quite some time ago with starting out looking at basic canonical resting fMRI networks and saying, okay, well, if we ping an individual network, can we get that whole network to kind of increase its connectivity? And while we're at it, why not test a theory about networks that are supposed to kind of be oppositional to one another, so task positive and the default mode networks, and see if we can show that antagonism directly in response to a TMS pulse. And so we were able to show that negative coupling between task positive and default mode networks, which was a great kind of causal relationship that couldn't be uncovered just from looking at resting fMRI alone. Another thing that we were able to show is that we could increase within network connectivity in response to these individual TMS pulses. So we can get the network, not just the place that we stimulated, increasing its connectivity when we're doing TMS in the scanner. And I should emphasize that this is not neuromodulation, right? We're not doing repetitive trains of TMS. These are single pulses interleaved with fMRI readouts to map circuits in the brain. So that's kind of a step along the way, I think, to more complex neuromodulation applications. So more recently in my lab at Penn, since I've been there, we're trying to find pathways down to subcortical regions that are involved in anxiety and depression. So we use a similar method where we're using this time individual fMRI data to guide where we apply TMS, and then we're doing the interleave TMS fMRI as the readout to show whether we can actually reach these deeper brain targets. And so our first stab at doing this is a sample of 15 healthy subjects. We stimulated multiple places for each of these putative pathways, the subgenual and the amygdala, and we're trying to see if we can show an actual response to in these deep subcortical regions that we care about. So these are common kind of fMRI-based abnormalities and anxiety and depression that are also the targets of treatment, subgenual cingulate and the amygdala. So those are our first two targets that we've tried so far. And we're actually able to show that we can influence the amygdala with TMS. So these are main-centered quantile averages from the amygdala, and we're showing that especially in a couple of subregions of the amygdala, centromedial and superficial amygdala, we're seeing an increase in the bold activity in response to a TMS pulse. So that suggests that we have found a pathway. And a student of mine, I just met with her this morning, is showing that in same subjects that we do an amygdala evoked response, we use the diffusion imaging and get some fiber density calculations from an amygdala network that's actually correlated with an evoked response to TMS. So we're pretty excited about that. The subgenual also being an important target. We showed in this initial study that we can move the subgenual in a negative direction, and not only the subgenual region of interest, but also a network distributed through the brain. That one is even more reliably kind of pushed off of its baseline. So we're pretty excited the network and the region itself are responding to TMS. Another thing that we did just to kind of get some additional indicators of specificity, right, with where we stimulated in the cortical surfaces. So this is all left hemisphere. I showed you guys the map of all the areas that we stimulated. And we're just looking at if we happen to be in a certain kind of major cortical parcel, would we have a stronger or weaker effect on the amygdala or the subgenual? Actually, when it comes to the amygdala, we found that if you stimulate in the inferior frontal gyrus or motor cortex, that you're getting a negative bold response, whereas if you hit this kind of superior frontal or anterior midfrontal zones, those are the ones that are really driving the positive bold response. So that suggests to us if we have a clinical target, like we want to potentially inhibit the amygdala, we might have a better chance of doing that in this inferior frontal or motor cortex. So that we're working on replicating that in a larger sample right now. Another thing that we're working on at the moment is a subgenual target. So it turns out that subgenual targets, the peaks that you stimulate, it actually doesn't matter very much. As long as you're hitting a high resting connectivity peak, it doesn't matter what prefrontal site you're stimulating it. And so it's less kind of important to choose a peak in a specific area. Another thing that we're playing around with, so these are very preliminary findings so far, is trying to determine the functional relevance of these new pathways that we're mapping. So one thing that we found, for example, is in depressed subjects, the evoked response in the subgenual cingulate is correlated with their negative affect from the penis. And so looking at that negative bold response in the subgenual, that's predictive of relatively lower negative affect. So that's very much in the direction that we would hope for. And it's sort of fortuitous that that relationship is coming out because we also are treating patients with PTSD and some in a small kind of trial with depression, and we're seeing some nice clinical responses. So in terms of the amygdala target, this is another preliminary set of data with an ongoing study where we bring them in and use the same kind of guided stimulation sites to do intermittent theta bursts, so an excitatory repetitive TMS actual neuromodulation through these pathways, and seeing if we can influence fear conditioning extinction in healthy subjects. So what we've found so far is that the amygdala pathway seems to be doing a better job at augmenting fear extinction. So this study is ongoing, but we're kind of excited with this preliminary data because if this is a pathway and a behavior relevant to PTSD and we can influence it, then that gives us a potential new target for treating that disorder. Another thing that I want to mention is that aside from the kind of straightforward, I think, clinical applications is just thinking about how TMS generally augments imaging experiments. So one of the shortfalls of imaging data on its own is that you can find a lot of correlated brain activity at rest or in response to a stimulus or a pre-post treatment, and it's hard to get a good understanding of whether there are specific kind of control nodes like John was talking about, or generally a hierarchy of communication in these brain areas. And so I think we're making some headway on these kind of questions, because neuroimaging traditionally hasn't been as clinically useful as I think a lot of us hoped for in decades past. But I think especially when it comes to TMS, where the brain data are a lot kind of closer to the actual treatment and how it's implemented, I think we have some real kind of leverage with the imaging data and for improving cognition and treatments for a variety of neuropsychiatric problems. And so getting some preliminary data like this, like John showed and like we're showing in our lab, moving behavior, symptoms, influencing the brain in these areas that have neuropsychiatric relevance, I think is a very promising kind of initial step towards personalizing and making brain stimulation more effective. So I've been very fortunate, especially with the Brain Initiative at NIH, to be funded for a lot of these projects. I have an excellent lab that have all contributed to the work that's that I've shown you guys, and exciting new applications. And with that, I will be glad to take questions.
Great. So let's open it up. I have not seen any questions in the chat window. So at this time, if anyone wants to unmute themselves and ask a question, let's go ahead and open the floor for that.
Hey, Desmond, can you hear me?
Yes, hi, Mike.
Hey, awesome work. I'm excited. I was taking pictures of my screen. Question for you. I'm a little surprised that it doesn't matter where in the dorsolateral prefrontal cortex you pulse for the subgenual suppression.
Yeah.
And it looks like you did the analysis in terms of parcels.
Right.
If you do the analysis in terms of kind of the the anti-subgenual blob and and then other stuff around it, or positive versus negative connectivity with the subgenual, do you see any signal at all, or does it seem like it's non-specific to topography?
So in that case, all of the sites that we included in that analysis had high functional connectivity peaks. So it's just in terms of within the sphere of a like canonical brain, where it doesn't make any difference if those peaks happen to be in one kind of major cortical parcel or another. So in terms of high positive or negative peaks, that's actually something I'm hoping we're going to figure out in the next couple of days. So we have enough subjects now to kind of split those apart. So the data I was showing are kind of a smaller pilot study, but now we've collected in upwards of 80 subjects, and I think we can start to split apart negative versus positive, but also they're just different pre-processing methods that we can go back post hoc and kind of reprocess the resting data in a couple of different ways, a couple of reasonable ways, and say, hey, are these better predictors of the TMS evoked response? So assuming that the evoked response is sort of like the ground truth of that pathway's communication, and then seeing if we can interrogate the resting data a little more carefully to see if we can figure out how to better predict the evoked response. So we're working on that very actively right now.
Awesome stuff.
Great. We're starting to get some questions come through the chat box. So let me read those out. All right, so I think that might be spurious noise. So, okay, so let me read this question. So great talk, Desmond. Could you speak to some of the trade-offs and technical hurdles to collecting fMRI during TMS?
Yeah, it's really difficult and really painful. So there are always kind of sources of extraneous noise. There's problems with the triggers. There's problems fitting the TMS coil into all the positions that you want it to be in and fixing that position. It's it's a full-time undertaking to collect good data. But there are specialized equipment, kind of manufacturers. MagVenture is the one that we use that has a MRI compatible TMS system. And so we use that, and it's works pretty well. We've the only custom equipment we've really had to design is kind of some integrating of the scanner and the TMS and being able to trigger them individually, and also to hold the TMS coil into the various positions that we want it to be in. So we've customized some of the equipment on that side.
Great. All right, I'm just going to take them as they come, but now I would like to open up the questions. So far, the last several have been for Desmond, but as we approach the end, if there are questions for either Desmond or John, I think that's perfectly fine. But the next couple are still following on your talk, Desmond. So although this one might be addressable by either speaker. I have come across certain studies focusing on functional connectivity in targeted regions of interest, resting fMRI, and electric field distribution in those regions, i.e. modeling, by TECS. I would like to have your opinion on that.
So I will say E-field modeling is a very kind of a well-used kind of tool in non-invasive brain stimulation. And I have done some calculations in my fMRI data. I haven't found it to be a strong predictor of the evoked response, but I'm still kind of messing around with different ways of categorizing the E-fields. So there's some single pulse electrophysiology data suggesting that the models for neural response may be a little bit exaggerated, and so maybe kind of tuning back our estimates of connectivities and playing around with those parameters, I think, is still something ongoing, especially for the single pulse data.
Couple more questions for you, Desmond. So first one, can you elaborate on the ITBS parameters in the fear conditioning study? So how is it targeted? How many pulses? Timing in relation to the acquisition and extinction? Are the effects only in the SCL?
Yeah, sure. So the ITBS protocol, we use the traditional Huang protocol. We do 2 rounds of 600 pulses, and the pulses are triplets. So we run them at, I believe in that one we did 80% resting motor threshold, and yeah, so that takes a pretty short period of time. We administer it to the amygdala seated functional connectivity target, which is where the IFG data that I showed. And so that's compared to the subgenual target. So we target those the same way in the scanner as we did in the navigated TMS behavioral session.
Great. Last written question for now. Desmond, I didn't quite follow with the negative bold and positive bold, its association with TMS stim and the dynamics of such hemodynamic responses. What about biphasic HRF, and how does this relate to specific cognitive stimuli and TMS stimulation?
Oh, wow, that's a lot. So, some of the ways we've been trying to better understand what the negative bold response means is by a collaboration with Michael Platt's lab, so they can do electrophysiology in the subgenual in response to single pulses. That part's we're still kind of cleaning up, but in response to like theta burst stimulation, and trying to understand what the negative bold actually means when we stimulate there. So I'd say that's still an open question in terms of the actual fMRI readouts, right? And getting a better look at the HRF. We didn't optimize our kind of spacing of our TMS pulses very well to capture an extended sort of HRF, but we did more recently in about a dozen subjects. We're spacing them out a little bit more, and we're going to try to figure out what kind of time bins and the kind of successive HRF dynamics are in response to the single pulses. So that's a work in progress. I hope to have an answer in the next few months.
Great. I'm going to take advantage of this little gap to ask you a question of my own, both of you. John, this is sort of practical. I know we're engaging it ourselves, and for Desmond, this might be a little speculative. So how are the methodologies and approaches that you articulate in your different talks impacted, or how would they need to be reconsidered in the context of individuals who don't have anatomically normal network distribution? For example, patients with brain lesions. Neither of you showed those specifically in your talk, so can you speak to that?
Yeah, so as Roy well knows, this is a favorite question of mine, and I guess I get more concerned over time in one way and less concerned in another. So I get more concerned in the sense that we're often using these sort of algorithms blind to anything that approximates ground truth, and then we're sort of subject to how good that approximates it. And there are a lot of very good studies now showing that we have false positive issues and some false negative issues in any major tractography I'm familiar with. And so I think we have that issue even before we reach the clinical sample, and I think it's still largely an open question the extent to which that's the same or a larger issue in clinical samples. So in that sense, I just think that there really is a need to, as Desmond was saying, making these things clinically actionable for folks to really understand those parameters, how they interact. If we can get postmortem and tracer studies to further and further refine those models, that seems just generically important for all of us. The thing I get more optimistic about over time is a case where, as you also see in Desmond's work, once we have some sort of handle on the anatomy we're talking about and the pathways that we think might matter, then we can start to constrain what we start asking in the first place, and then use what look very much like prospective enrollment designs. So, it still remains to be seen for our own particular pilot shared with Roy for these particular measures. But I'm optimistic that, in the end, the cognitive neuroscience and the behavior is really our guide here for those of us that care about those outcomes. So that tends to be how I think about it at this moment. Desmond, I don't know if you feel the same or differently. I'd be curious.
Yeah. I would sort of punt this clinical question to Roy if somebody gave it to me, because I'm thinking, all right, the rehabilitation process in somebody with a lesion, an event, a neurological event is sort of tricky and time dependent. And for Roy and other people who have studied that, I'll sort of concede to them that there's definitely critical time periods where different kinds of interventions are going to sort of have better effects. And I think it's really a cool area of doing more work on. And I would love it if somebody like that wanted to team up with me to measure TMS fMRI through the circuits where you might be doing a rehabilitation and see how they change at different time periods in the rehabilitation.
Wait a second, we're somebody like that. All right, well, that's great. Listen, I see some questions that are actually being, Desmond, it seems like you're responding to people in the chat box, which is great. We are at the top of the hour, so we're technically out of time. So I guess anyone, you can continue to respond. We can maybe leave the window open, but thank you everyone. Thank you, John and Desmond, for speaking today, and for everyone else for attending. I thought it was a great session. Have a great rest of your day.
Yeah, thanks everyone.
Thank you, everyone.